Hello everyone, this is Nunzio from the Department of Pharmacy and Pharmaceutical Sciences of the University of Bari. The University of Bari is one of the oldest Italian state university and is part of a consortium, namely the Consorzio per Valutazioni Biologiche and Farmacologiche, CVBF, which is a not-for-profit organization founded in 2000 with the mission to perform research and education in the pharmaceutical field. CVBF coordinates today the European Paediatric Translational Research Infrastructure, also known as Sept, which aims to create a framework for a new research infrastructure intended to enhance technology-driven pediatric research in drug discovery and early development phases to be translated into clinical research and pediatric use of medicines. As underlined by Sept, children are not small adults, so it is necessary to develop suitable formulation for the different pediatric age categories. In particular, children with an age between 2 to 11 years old represent an important part of patients affected by inflammatory bowel disease. Current treatments involve the use of corticosteroids, including budesonide, that is a synthetic corticosteroid with high anti-inflammatory potency but a very low bioavailability and systemic activity when administered orally. Specific colon drug delivery systems are of great interest for the local treatment of the inflammatory bowel disease. Therefore, the purpose of this study, which has been outlined in this poster, was to produce with the innovative prilling technique an oral multiparticulate of budesonide for the treatment of the inflammatory bowel disease in children. These drug delivery systems have to be able to target the colon to permit an accurate and flexible dosing and also to enhance patient compliance. In particular, as shown in the figure, for the realization of microparticles, a 2% weight by volume sodium alginate solution has been used. Aqueous dispersion of eudragite polymer, a pH-dependent polymer that provides a colon-specific release, was added to this solution, obtaining 4 mixtures with different ration of eudragite and also alginate. Then budesonide was added as suspension or as solution if added as inclusion complex with hydroxypropyl beta-cyclodextrin. Furthermore, microparticles were prepared starting with the same budesonide suspension with the addition of inulin, an enzymatic dependent polysaccharide that is not digestible by humans but is used as a nutrient source by bacteria residing in the colon. The resulting preparation were processed by prilling technique using the encapsulator equipped with a single nose. This technique can produce microparticles by breaking apart a laminar jet of polymer solution into a row of mono-sized drops by means of a vibrating nozzle device. The resultant droplets fall into a consolidation medium in which they are solidified as beads. In this study was used a solution of calcium chloride 0.3 molar as consolidating bath. As shown in the tables, the microparticles were obtained with high yields, homogeneous size in line with the nozzle used of 450 micrometers, and high encapsulation for the formulation with budesonide and low encapsulation efficiency in the presence of the complex hydroxypropyl beta-cyclodextrin and budesonide. Also, in vitro release study has been performed by using a specific buffer simulating the gastroenteric environment. In summary, oral pediatric microparticulate formulation of budesonide based on alginate, eudragit, or alginate, eudragit, and inulin were developed using the prilling technique. The former achieved a good results in terms of flexibility and release to colon, but the latter, having a mixture of both triggered pH and enzyme-dependent polymers, could provide colon-specific release of budesonide with interesting release profiles more suitable for the treatment of the inflammatory bowel disease in children. Thank you for your attention.